to Study Life. The name of our chapter is Enhancement in Food Production, and in that, the topic which we are seeing is plant tissue culture. Before seeing plant tissue culture, let us see a unique property which takes place in plants. Now, plants are such special creatures that they have the ability of cellular totipotency. What is cellular totipotency? It is the ability of a single plant cell. A single plant cell can divide and differentiate into a complete new plant when it is kept under suitable environment conditions, which means when we have a single plant cell, from that single plant cell, when we keep it in a tube with a nutrient medium, it has the ability to divide and differentiate, means it is going to mature and it is going to give us a complete new plant. That is called as cellular totipotency. So, only plant cells have this ability. What about animal cells? Animal cells do not have this ability because animal cells are pluripotent. Pluripotent means only one single cell cannot form the entire organism in animal body except for zygote and blastocyst. Otherwise, plant cells only are totipotent. What is plant tissue culture? When we see plant tissue culture, it is the culturing or growing of, see, the name culture itself suggests us that we are going to culture or grow. It is the culturing or growing of, either we take isolated protoplast of cells, which means here we are taking only the protoplasm of cells, isolated protoplast of cells, or cells or tissues on nutrient medium, where we are going to take them on nutrient medium under aseptic conditions, on nutrient medium under aseptic conditions. Now, aseptic conditions means sterile conditions to obtain entire plant or plant parts. Let us see what I said. I am saying that we have this tube. In this, we will be having a nutrient medium and we would be culturing or growing isolated protoplast of cells or tissues where on a nutrient medium, this is our nutrient medium, under aseptic condition. When we say aseptic condition, we mean here that it is a sterile condition, which means it will be covered on the top by the cotton. So here we are going to take aseptic condition, which means here we have sterile conditions. So this was plant tissue. So this is our aseptic condition. So we are getting an entire plant. So this is going to form an entire plant or plant parts. So here we would be having our plant or plant parts. This is called as plant tissue culture. So let us see who discovered this and what are the requirements of plant tissue culture. So this plant tissue culture was discovered by the scientist known as Haberland. Haberland, he in the year 1902, he described about plant tissue culture and he first explained this topic, which is plant tissue culture. And he has given us the example of cellular totipotency. So all the credit goes to him, Haberland, in the year 1902. So let's see what are the basic requirements for plant tissue culture. For plant tissue culture, our first requirement is we require the explant. What is the explant? We would be seeing in the next session. Explant. Second is we require aseptic sterile nutrient medium. We require nutrient medium as per the requirement. The third requirement is controlled laboratory conditions. In this controlled laboratory conditions, it includes maintenance of temperature, humidity and light. So controlled laboratory conditions. Then the fourth requirement is plant growth hormones. Which hormones are required? The hormones like auxin and cytokinin are required. Now, why they are required, we would be seeing later. Hormones like auxin and cytokinin, they are required. So, these are the main requirements of plant tissue culture. And the fifth and the most important one is aseptic laboratory conditions. So let us have a brief look at what are the requirements of plant tissue culture. These are the requirements of plant tissue culture. First is the explant. Second is nutrient medium. Third is controlled laboratory conditions, which includes temperature, light and humidity, hormones like auxin and cytokinin, and maintenance of aseptic condition, which is sterile conditions. So these are the requirements. Now let us see how nutrient medium is prepared. Now when we say about the nutrient medium, this nutrient medium can be made as per the requirement. It can be designed as per the requirement. When we say nutrient medium, it contains variety of organic and inorganic nutrients. Organic and inorganic nutrients like it contains sucrose, vitamins, it contains hormones as we have stated over here, which are auxins and cytokinins. The pH of the nutrient medium has to be maintained between 5 to 5.8, which is slightly acidic. Absolutely correct. So pH has to be maintained between 5 to 5.8. So suppose we take water in a vessel. We add these nutrients, sucrose, vitamins, hormones, different salts. We mix. What do we get? We get liquid nutrient medium. But we have to make the medium semi-solid, which is jelly-like. To make the medium semi-solid, we add we add agar-agar, which is a polysaccharide, which is obtained from red algae. This agar-agar polysaccharide is obtained from an algae which lives in the ocean. So when this is added, the medium becomes semi-solid. Which medium had Haberland used? This is a very important point. Haberland had used Knoop salt solution and sucrose. When Haberland had done this experiment, he had used Knoop salt solution and sucrose as a medium for plant tissue culture. What about nowadays? Nowadays, the medium which we use is called as MS medium, which is Murashige and Skoog medium is used for plant tissue culture. So this is about the nutrient medium. Next important requirement is this aseptic condition, which includes sterilization. Now sterilization is one of the most important criteria for our plant tissue culture, because if there is any minor contamination, it will result in experiment to fail by growth of harmful bacteria and fungi. So sterilization is one of the most important criteria. This sterilization has to be done for all the things. All means it has to be done for the nutrient medium, of the explant and of the glassware. Of the explant and of the glassware is done. So sterilization of all the requirements is absolutely necessary. The question comes, how does this sterilization is performed? This sterilization can be dry sterilization, it can be wet sterilization, dry or wet sterilization. Dry sterilization is done in microwave oven. Wet sterilization is done in an instrument which is called as autoclave, which resembles the pressure cooker, or it can be alcohol based sterilization. This alcohol based sterilization is done for the tables and person who's handling. So they use alcohol to sterilize their hands or tables. Now, dry sterilization, wet sterilization, alcohol-based sterilization. For sterilization of the explant, the most important question, our explant, which is the material which we are going to use, This explant has to be sterilized by using 70% ethyl alcohol or 10% hydrogen peroxide or using 1% sodium hypochlorite. So either of these substances can be used for sterilization of the explant. It is 70% ethyl alcohol, 10% hydrogen peroxide or 1% sodium hypochlorite. So this was all about plant tissue culture, from where we can obtain an entire new plant by just taking a small part of the plant, which is the explant. For more videos, please stay tuned to Study Life. Thank you.